Hey everyone, Jen Bailey here again with AP Bio Walkthroughs. This is test A from the, From the Princeton AP Biology Premium Prep from Princeton Review for the year 2021. You can go out, get you a copy if you don't have it already, register it. But if you don't have one, that's okay, because we are going to look just at this picture, just at this question. And so you don't necessarily have to have the test to follow along. Okay, so this is the last question on this exam, which is kind of fun. See there, end of exam. And so this is the last short answer. So you will have about 11 minutes to do this. Okay, so let's go ahead and jump right on in. The fruit fly, Drosophila melanogaster, is an ideal organism on which to study genetic mechanisms. The organism has simple food requirements, occupies little space, and the reproductive life cycle is complete in about 12 days at room temperature, allowing for a quick analysis of test crosses. Interesting information, a bit superfluous, perhaps, we shall see. In addition, fruit flies produce large numbers of offspring, which allows for sufficient data to be collected quickly. Many Drosophila genes are homologous to human genes and are studied to gain a better understanding of what role these proteins have in humans. So basically, they gave you some background there. Some of that stuff you should already know, being an AP biology student. Okay, so they're just getting you oriented there. They mentioned test crosses right here. We should automatically be thinking about our squares, right, and our alleles and how we can combine them in dihybrid crosses. Okay, so that should click on. To understand this inheritance pattern of certain genes in Drosophila, the following experiments were carried out. Assume that the alleles for red eyes, brown body, and normal wings are dominant. That means we're going to write them as capital, right? And the alleles for white eyes, ebony body, and vestigial wings are recessive. Okay, how do we keep that in our minds? The first thing I would do, let's go ahead and write it down on our problem here, okay? And so I'm going to go ahead and make my own code for this, and this is something you can do. All right, so when we're assigning these, we can, pretty much use any code we want that will help us remember. I'm going to go ahead and start writing. So let's say, okay, so we've got the dominant alleles, right? We've got red eyes. Let's call that capital E, okay? Put red eyes. And then we have white eyes. Okay, let's make that recessive. Let's put white eyes. Okay, let's see what else is dominant. Okay, we'll do B for body. So that'll be brown for body. And then little b is ebony or black. Okay, and then what's the other one? Wings. So capital W will be for normal wings. Okay, and little w will be for vestigial. Okay, there we go. All right, so now they give us some experiments. Okay, so let's check out these experiments. Experiment 1, a red-eyed female and a white-eyed male were crossed. The subsequent generation of flies was also crossed. The phenotypic results of both generations are shown below. Okay, so we've got a red-eyed female. That means she's probably either going to be what? Heterozygous or homozygous. 2 capital E's. Okay, we don't know yet. We might have to find out at some point. A white-eyed male, what's that going to be? EE. Okay, so they were crossed. The subsequent generation of flies was also crossed. The phenotypic results of both generations are shown below. Okay, so we could pause and do that little cross really quick. It wouldn't be that difficult, but we have to do 2, right? So let's just kind of make a note. All right, so there's our female and there's our male. Okay, so we might end up doing that for experiment 1. Okay, so experiment 2, 5 of the red-eyed females from the F1 generation up here were brought down right here. So there's the 5. Okay, when I first read that, I didn't understand that these were the same, but I did the math and it works out, yeah. So that's the F1 generation. So 5 of these guys were brought down and mated with white-eye males. Okay, so we've got 5 of the red-eye females mated with more white-eye males. Again, it's gonna look like either the heterozygous or the homozygous dominant for those red-eye females, and it's gonna be the little e's for for the white-eyed males. Okay, all right. So we could also use this for experiment 2. Okay, let's go down. Let's go down to experiment 3. A male heterozygous for body color and wing type is crossed with an ebony vestigial winged female. The results of the cross are shown below. Okay, so we can look at those numbers in a second, but let's think about what the alleles will be for that. A male heterozygous for body color. What's that going to be? That's going to be, I'll go ahead and do my male figure right here. That's going to be heterozygous for body color and wing type. Okay, so we've got something that looks like that. Was crossed with an ebony vestigial winged female. Okay, so we've got heterozygous male crossed with a recessive female. Okay. All right, there we go. The results of the cross are shown below. We don't need to worry about that quite yet. Let's go ahead and jump to the first question here. All right. There we go. Identify the percentage of flies in the F2 generation in experiment 1 that were homozygous recessive. Okay, so it's saying what? Identify what? The percentage. The percentage, guys, not the individual number. So let's go up to the F2 generation in experiment 1, which is going to be right here. And how many of those are homozygous recessive? Well, we know the red eye. If you've got red eyes, then you're either homozygous dominant, right, or you're heterozygous dominant. So it's not going to be any of the red eyes. It's going to be the white eye males. We've got 14, okay? So that's how many individuals we've got, 14, but they want the percentage. So what do? We just add up these numbers. That's 53, okay? Oh my goodness, I'm not really sure what happened just there. Sorry, guys. That's 53, okay? And we divide 14 by 53. And that is going to be 26.4. I'm having some trouble with this. There we go. Okay, and so that you can do that math really quick on your calculator. It's 26.4%. Okay, that is going to be the answer to A, 26.4%. All right, so let's go ahead and erase what we can over here. We already know about that. All right, let's look at number 2. Describe, or letter B, I should say. Describe the difference between the red-eyed female in experiment 1 and the red-eyed female in experiment 2. Aha, so since we don't know whether these females we don't know whether they were heterozygous or homozygous for red eyes, that's what we got to figure out. And we can do that, we can figure that out by the results, okay? So the first thing that we need to do is we got to do our cross. All right, so let's go ahead and do this. So I'll do a blue. All right, so I'll bring down the E here and the E here. We've got a heterozygous for red eyes. Oh, but right here in our cross, we've got a homozygous for white eyes. Right here, we've got another heterozygous and a homozygous. Okay, so this looks like 50-50 if this female right here is heterozygous. We get 50-50 for red-eyed females, or sorry, we got 50-50 for red eyes and white eyes. But look what we have over here. Do we have any white eyes at all? No, we definitely do not have any white eyes. So it's probably going to be the homozygous dominant. Let's go ahead and do that cross and we can check. All right, so we've got a heterozygous You got another heterozygous. We've got another heterozygous, and we've got another heterozygous. They are all heterozygous. And if we look back here again, that means that we should have all red eyes and 0 white eyes. And look, that's exactly what we have. We have all white eyes and 0, or all red eyes and 0 white eyes. So we know that this female is homozygous dominant. Okay. Not sure if you guys are seeing that weird glitch that's happening where it takes me out, but I sure am. All right, there we go. So in order to find out what combination of alleles this F1 generation is in experiment 2, all we need to do is look back to the heterozygous cross that we did right here and look at the math. And as we saw before, it's a 50-50 split, right? So we should see something like a 50-50 split over here, with 50% being red eyes and 50% being white eyes. So let's see, is that what we see? In the first generation, not really. In the second generation, look at this. Oh, that's right, because the first generation is this exactly. Okay, so that's not the cross, that first line there. The real cross is the second line. Okay, so we should be looking for math on here. And yes, we've got a red eye and a white eye and a red eye and a white eye. Not perfect, right? But we've got 8, 8, 9, and 8. That's a pretty close percentage to 50-50. So I'm going to say that this girl over here is heterozygous. Okay, so that's going to be my main answer for this one. All right, all you've got to say is the female in experiment 1 is homozygous for red eyes based on the percentage or based on the fact that when you cross them, you get all red eyes. You get 0 white eyes. Okay, and so then when you're explaining about the second female in experiment 2, you're going to say that, due to the 50-50 split between white eye and red eye, the female is most likely the heterozygous form, okay, with 1 dominant allele for red eye and 1 recessive. All right, but that dominant E takes the cake there. Okay, so the next one we're going to do is C. All right, so assume that the gene for body color and wing size are on the same chromosome. Calculate the recombination frequency in experiment 4. Okay, so if you do not know what recombination frequency is, that is okay. It is very simple. All right. We're going to go ahead and I'll go ahead and just write it right here for you so you can take this down. First of all, what in the world are recombinants? Recombinants are any of the offspring that don't look exactly like the parents. Okay, so for this one, we crossed a brown, okay, brown and normal wings, okay, something that looks like B and W, right here. We crossed that guy with a recessive girl with a black body, ebony body, and vestigial wings. Okay, so any recombination of these traits down here in the table is going to be one of our recombinants. So what in the world does that mean? That means if we have like a brown body with vestigial wings. See, that's 2 different traits that either of the parents have, but none of them both have it. So basically what you're looking at there is evidence of crossing over, and we'll talk about that in just a second. But first, it wants us to calculate the recombination frequency. Also, they say experiment 4. There is no experiment 4. That is a typo. There is also a typo in the answers up here. It adds these all up to 51 when it's really 53. So every now and then you'll catch those typos on these practice tests. But anyway, everyone's human, right? So the recombinant frequency is the number of recombinant offspring. All right, so any of those offspring that have different combination of traits than their parent divided by, whoops, just pretend that's a nice straight line there. divided by total offspring. Okay, and then you multiply that by 100, and that's going to be the frequency. All right, that'll be the percentage. Okay, so we can easily do this. Oops. So if we add up all of the numbers in this column here, in these columns here, you know, you can do that on your own for fun, but I already did it, and it's 130. Okay, so the total's 130, so I put that on the bottom. Make that a better one. All right, and now we find the total number of recombinants. Okay, so first to do that, I look for these and I mark them out. Okay, so a brown body and normal wings. Goodbye. And then an ebony body and vestigial wings. Goodbye. Okay, then that means we're just adding these numbers up. You get a 7. Okay, 7 divided by 130 times 100%. This equals 5%. Okay, I have 5.4. You can round that down to 5%, all right? But I think they'll take it if you give it a couple of decimal points. They don't necessarily say, you know, to round or anything. Okay, so that is going to be your answer for C. All right, so let's do D. Okay, so I'm going to go ahead and do all this. All right, so when we look at D, it's asking us a general question. Okay, I think we're all done with our calculations. It's saying explain why gene linkage is an exception to one of Mendel's laws. Aha. All right, so we need to draw on our knowledge of biology and explain why this recombination stuff that's going on here is an actual exception to one of Mendel's laws. First of all, which one of Mendel's laws? You can think back, but automatically you should probably think of independent assortment, right? Because these genes are not necessarily sorting it independently all the time, as shown by this recombination table. Okay, you've got some that are recombining with each other. Okay, so what in the world does this mean exactly, and where does this happen? How are these recombining? Well, remember when you've got your sister chromatids, okay, and they're linking up with the other chromatids, you get crossover. Okay, so the site of crossing over is where the recombination can happen. All right, and this whole thing right here, crossing over, is the reason why we have so much variation, right? It's why sexual reproduction is more prevalent than asexual reproduction, because it gives you more cards to play with, right? It shuffles your genes. It can mutations can happen with all of the stuff that needs to happen in meiosis. And so this crossing over event is where recombination can happen. All right, so when you're explaining why gene linkage is an exception to one of Mendel's laws, you're going to state it is an exception to the law of independent assortment. And here's why, right? Because sometimes when you're crossing over right here, you can actually have ones be really close to each other, right? And the physical site of crossing over might mean that more than one gene is switched. Okay, it's not always like that. As you can see, you know, this is only 5%. Recombinants are always going to be under 50%, FYI. But you don't need to necessarily explain all of that here. What you don't want to do is brain dump right here, right? You want to get in and get out. So all you got to do is explain why gene linkage is an exception to one of Mendel's laws. We've just explained that because crossing over can sometimes incorporate more than one gene. Thus, recombination is an exception to the law of independent assortment. Okay? And then, woohoo, you're done with your test.